So here's our second part of the lecture, the origin and diversity of life. Keep in mind, based on the scientific evidence, the Earth is about 4.6, 4.8 billion years old. Very, very old Earth. And it's believed life originated 3.8, maybe 3.6 billion years ago. How it got here, though, is being tested. There's several hypotheses that are currently being tested, and our goal is to figure out what is the best scientific explanation. But once life got here, it started to change. It adapted, it evolved, and it evolved into the diversity of life we see on Earth today. And the sedimentary rock layers tell us a great history of the Earth. We can look at this progression of evolution when we look at the rock layer. Now, the reality is not everything is going to be in the rock layer. Not everything is going to be present in a fossilized form. So there will be things that we have to make assumptions on. We have to make inferences. And then we continue to look for that evidence to see, are there other ways to support that progression of life or that evolution of life? And kind of filling in the puzzle as we go and we continue to do more and more research. So a lot of the past can be reconstructed from the fossil record. What we do know is the earliest, the first cells are, that we have evidence of today. This could change in the near future, but the first cells that we have any evidence of are these little things called microfossils. These are basically fossilized forms of microscopic life. The oldest known to science date back 3.5 billion years. Alright, so that is the oldest known evidence of life on Earth. More research, maybe we'll find something older, but currently this is our oldest physical evidence of life on Earth in the fossil record, these microfossils. Now, when we age fossils, we use two different techniques. The first one is called relative aging. The position of a fossil in the sedimentary layer indicates approximate age. So that's perspective. If it's sitting in the fossil layer so many feet down, so many centimeters down, etc., it can give us a rough age, a relative age of that fossil. Say, well, it's approximately 12 million years old or 375 million years old, etc., based upon its position in the fossil layer. Quite often we use index fossils to help ID the relative age of other fossils. So there are certain fossils that we only find at certain periods of the Earth's history. They existed for a window and then they disappeared. Those are index fossils. So other fossils that are present in the same rock layer, the same sedimentary layer, would assumably be the same age. Now, they may have been there earlier or may have existed longer, but if they're side by side in a fossil layer, we know, hey, they're around at the same time. The other technique is what's called absolute age, to determine the age of a fossil. This is estimated by using the rates of radioactive decay of these isotopes. So when we look at different isotopes, scientifically, the chemists have figured out how long does it take for it to break down into what's called the half-life. So it goes from a certain amount to half that amount in so many years. And then the same amount of years reduces it in half again. And then the same amount of years in half again, and in half again, and so on, and so on, and so on. So carbon is really one of the main ones that's used for the radioactive decay or the isotopes. Is it carbon-12? Is it carbon-13? Is it carbon-14? What is the half-life? So making it just simple numbers, if we were trying to study the half-life and we had 100 units of carbon starting out, it would take 5,700 billion years to reduce it in half to 500 units of carbon. Another 5,700 billion years to reduce it in half again to 250 units, and so on and so on. So if you grab a handful of carbon and you go, wow, there's 85 parts here, you can do the math and mathematically determine the age of that fossil or age of that sample that contains that volume of carbon. So it's a way to dial it in a little bit more specific than just relative dating, where we're getting a rough picture. Absolute aging, absolute dating gives us a much finer picture of time when we look at it from this perspective. Okay, so here's an example looking at the amount of the parent isotope. That's the purple, and it takes so many years to get reduced in half. The same amount of years to get reduced in half again, the same amount to reduce in half, and the same amount of years to reduce in half again, and so on and so on. So this is a progression of decay on this, what we call this linear slope here, and we know how many half-lives it takes to do this. So we know the length of a half-life. If we have this amount of the isotope present, 1/16, we can extrapolate backwards, add up the half-lives it would take to get to that point, and tell us how old that specimen actually is. So again, this helps, but it does have limitations. All these dating techniques have limitations, but it gives us a clearer picture. We don't rely solely on one technique or one piece of information when we're working with the fossil record and evolution. It's just one piece to support the bigger picture. There's lots and lots of other pieces that we work with as well. Okay, so that earliest fossil evidence, we're guessing and estimating, 3.8 billion years ago is the assumed, or 3.5 billion years is the estimated age for those first cells. Now, the first eukaryotic cells, these are cells with a nucleus. Okay, so now we're getting away from prokaryotic. Cells with the nucleus, these are estimated to have arisen or become present first about 1.5 billion years ago. All right, so that is our estimation on the eukaryotic cell, the cell with the nucleus. How did it get here? When did it first show up? About 1.5 billion years ago. So life existed on Earth in single-celled form for almost 2 billion years before we start to see eukaryotic cells. And the eukaryotic cell, is the thought is it originated through a process known as endosymbiosis. Okay, endosymbiosis is, let me get this font, is the incorporation of one cell into another. So the thought is early bacteria, one of them engulfed another one. It engulfed another single-celled organism, and instead of destroying it, killing it, eating it, it started working together. They started working in conjunction and improving the success or the survival of both organisms, and they began to become mutualistic with each other, supporting each other, benefiting each other. So this is how it's thought mitochondria came to be inside of eukaryotic cells. This is how it's thought the chloroplast came to be. They were engulfed. They were cyanobacteria and then engulfed by another cell, and then they evolved together and created those organelles within eukaryotic cells, giving rise to different lines of algae, green algae, red algae, etc. So when we're looking at this, endosymbiosis process, this has been replicated in the laboratory, taking single-celled organisms and incorporating another single-celled organism into it, and then they become mutualistic. They work together. So this has been pretty well tested and shown to be possible. So it's thought, again, this is how the eukaryotic cells first originated. So when we look at eukaryotic cells, we have some special characteristics, certain key features and characteristics that make us a little bit different. Compartmentalization. Make sure I spell this right for you guys. Compartmentalization is a feature of eukaryotic cells. We have all these little pieces and parts and components inside of our cell. We call them cell organelles. Mitochondria, Golgi, endoplasmic reticulum, lysosomes, etc., etc., etc. Those are a big feature of eukaryotic cells that make us a little bit different than the prokaryotic cells. Not all, but lots of eukaryotic cells exhibit multicellularity. We're multicellular organisms. Not all, but a lot of eukaryotic organisms are multicellular. So then those cells had to work together. Prokaryotic cells, individual, single-celled organisms. And we will see sexual reproduction in eukaryotic cells. It's different than the prokaryotes. They exchange genetics, but we don't see true male-females in bacteria or the archaea, but we do see this in eukaryotic cells. So again, some differences between us and the prokaryotic cells. Having these features leads to the increase in genetic diversity, especially sexual reproduction. And that increase in genetic diversity is a key feature for evolution to occur with. Okay, so an example of how it's thought eukaryotic cells originated and became multicellular is this little slime mold, Dictyostelium discoideum. These are single-celled organisms. They're eukaryotes, single-celled, but they will do a colonial aggregation. In times of stress, they group together and form a multicellular organism. So if the environment's changing, food's depleting, pH is shifting, temperature, moisture, etc., they come together and, like these little crazy transformer things, they group together, they attach to each other, and then they start working in conjunction as a multicellular organism. So we can see this with these things, and it's thought this is what possibly led to multicellularity in other eukaryotic cells, this type of scenario. The sexual reproduction leads to greater genetic diversity. Consider the process of meiosis. You know, we talked about spermatogenesis and oogenesis. Both of those lead to genetic diversity, and especially the crossing over process that occurs during prophase 1 of meiosis. Huge genetic diversity results from that. This is why whenever you have multiple children from the same two parents who reproduce sexually, they are not identical because of crossing over. So you have so many billions and billions of combinations that give us such a great genetic diversity. It's amazing when we look at sexually reproducing organisms and the genetic diversity they can produce. Okay, so all these things helped drive the evolution of the eukaryotic organisms. Prokaryotic organisms are still evolving, still changing. We can see those in front of our eyes. But it tends to be the eukaryotes that we're more interested in because they're the big, more interesting organisms. So what we'll look at in our next lecture are the major innovations that allowed life to adapt to land. It was a challenging environment that we overcame.